So without further ado, I actually would like to invite my friend Udai Pandey, and he will be giving us a talk about his recent achievements and his recent discoveries. And he had one paper accepted to Nature Communications recently. So Udai, please welcome. Can you help us change the slides? Can you help us change the slides? We need to change the slides. Oh, it's already changed. Perfect.
Good morning, everyone. Thank you so much for coming, and thank you very much to my friend and colleague, Pandey, for organizing such a wonderful symposium. And I am honored to be here, and I would like to talk about our recent work that we started several years back at Louisiana State University, and then we continued working on this at University of Pittsburgh Medical Center. So I am not going to spend too much time on giving background information, but just to, I made these slides so that I can remind myself what is this disease and what are we working on. And this disease was popularized by this New York Yankee, Lou Gehrig, who was diagnosed with this disease at the prime of his career, and he could not continue his sports career because the disease affected him so badly. There are other players like Steve Gleason in New Orleans. I have had an opportunity to see him, and that affected him. He was diagnosed with ALS, and this disease affected him so badly. So this is a disease caused by progressive loss of upper and lower motor neuron, and generally patients, they survive from 3 to 5 years after being diagnosed, and this is a fatal neurological disease. And there are genes, there are two, it can be classified in two parts. One is a familial form of ALS, a sporadic form of ALS, and about 10 to 20% of patients, they have a familial form of ALS, whereas about 80 to 90%, they are sporadic in nature. And it's important that these familial and sporadic cases are clinically indistinguishable. They have same clinical presentation, same pathological features. So there is a great deal of similarity between these patients. And these are genes. This is just like a slide that I liked. And first gene that was discovered and linked with ALS was SOD1. And since then, there have been a series of genes that has been identified. So my lab started working on one of the gene, ALS-causing gene, which is called fused-in sarcoma. And this gene was discovered in 2009 by two different groups. And we developed cellular and animal models of FUS-mediated ALS, as well as several other genes, which has been linked with ALS pathogenesis. So we have models of all these ALS-causing genes, and we perform genetic screens as well as small molecule screens in our lab using these models. And we try to understand what goes wrong because of pathogenic mutations in these genes. So again, just we talked, ALS can be classified in two different parts. And one of the features that you see is TDP43 pathology. About 97% of ALS patients, they show TDP43 pathology. And there are genetic components, whereas like Pandey talked, there are veterans who are more likely to affected by ALS as compared to general population. So I will be talking a little bit about some of the external factors that are linked or they are predisposing to neurodegenerative diseases. So again, the point that I wanted to make is they are very similar. So my lab started working on this protein, which is called fused-in sarcoma. And this is an RNA-binding protein. Pathogenic mutation in this protein leads to juvenile and aggressive form of ALS. And this protein is very similar to another one, which is called TDP43, and this is predominantly nuclear protein, which is involved in regulating different aspects of RNA metabolism. And because of pathogenic mutations, a tiny fraction of this protein becomes mislocalized to the cytoplasm, where it sequesters into cytoplasmic granules, and it becomes aggregated. So this is the original discovery that was made, and you can see because of a pathogenic mutation, which is P525L, protein becomes mislocalized to the cytoplasm. So my lab has been using a series of cellular and animal models to understand the mechanism of ALS, or these RNA binding proteins, and how pathogenic mutations in these proteins, they cause symptoms that we see in patients. So we are using Drosophila as a model system, as a complementary model system in conjunction with other model organisms to understand the molecular mechanisms. And there are many advantages of using Drosophila as a model organism. The genome is sequenced, so we know how many genes are in there in Drosophila, and there are large collection of tools available. That means we don't have to develop our tools to study any human disease in Drosophila as well as in other model organisms. We can perform genetic screens in this model organism. And I will give you an example of genetic screen that we performed. And fruit flies has been used for more than 100 years, and many important discoveries has been made, including circadian rhythm, sleep cycle. That was mapped in Drosophila, and a Nobel Prize was given a few years back to fruit fly researchers few years back. And fruit flies, they live approximately 60 to 80 days, and that means 1 day of human life. You can compare with 1 year of human life is 1 day of fly life. And about 75% of human disease-related genes are conserved in Drosophila and human. So that makes this model organism more attractable. So when I started this project, we took a normal version of fused in sarcoma and then 3 different mutant versions. And we targeted expression of these proteins to Drosophila eyes. And this is normal Drosophila eye, which is made up of 600 to 800 ommatidia. And you can see they have high architecture structure. And this is part of nervous system. So when you express normal or mutant version of first protein, you see different degree of degeneration. That means there is loss of tissues, there is a loss of pigmentation, necrotic patches, and you can easily observe it under the scope, and which becomes more obvious if you perform a scanning electron microscopy. And in high power, you can see how much these eyes are messed up, suggesting that these neurons, they do not like having this protein, especially mutant version. And again, it becomes more obvious if you cut sections of these, and you can see degeneration of tissues, loss of architecture. So then, since it is a neuronal disease, we targeted expression to Drosophila neurons. And we found that these animals, they do not reach up to adult stage, specifically these 3 mutant version. Wild type has also toxicity, but not as strong as a mutant version. We took early stages of these transgenic animals, and we allowed them to crawl on agar plate, and we measured how fast they are moving to look for their motor function. And we found that these animals expressing mutant version of FUS, they have larval paralysis, which suggests that it recapitulates features that we see in patients who carry this mutation. So then next thing we did was we looked for the subcellular distribution of this protein. And this is just like control, and we stained it with lamin. Anything inside is nuclear, outside is cytoplasmic. Wild type FUS, as expected, was predominantly nuclear, whereas mutant was distributed in nuclear and cytoplasm. So this is a long story. We had a model. So what can we do with this model? So one of the power of this model organism is we can do unbiased genetic screen. And we performed a genetic screen that was done in 2 steps. First, we did a rapid whole genome scan, and then we identified some candidate intervals, and then we decided to map it. So in Drosophila, there is a library available, and Drosophila has 4 different chromosomes, just like in normal flies, and there are deficiency lines available that has unique deletion. And these deletional points are molecularly mapped. So that means we know how many genes are deleted from point A to B. And there are small deletions and large deletions. So if you have a model that shows a phenotype, you can take this model and cross it with your deficiency kit. So using this approach, we identified enhancers and suppressors of first toxicity. That means we identified some genetic intervals where there are certain genes which might enhance or suppress toxicity. But there are 10 or 20 or 30 genes in this interval. So we are in a process of mapping these genes. And 2 of the genes that we were able to successfully map, one that I'm going to talk about here, and another one, my postdoc fellow will be presenting a poster on that. So feel free to stop by on her poster. So, so far we have about 17 strong enhancers, 17 suppressors and 8 enhancers. And when we looked up in these intervals, there are interesting genes that are relevant to neurodegenerative diseases, including genes which are responsible for stress granule dynamics or RNA metabolism. So one of the genes that we were able to successfully map was on the chromosome number 2. And this deficiency line that we have on chromosome number 2 showed a strong suppression of first toxicity. So we started using a small deletion point, and then we narrowed it down, and using RNA line to specifically target genes which are in that interval, we were able to map one of the gene which is called muscle blind. And I'll talk more about muscle blind, but we found that muscle blind is the only one that was able to suppress toxicity when we depleted it individually, not the other genes. So we tested our lines, we did extensive characterization of suppression effect, not using the eyes, but also neuromuscular junction, also locomotor function, lifespan, and so many, a battery of tests that we ran on this to make sure that muscle blind is a real modulator of first toxicity. So what is muscle blind? This is an RNA binding protein that is highly conserved across the species, suggesting that it has some function that is required from lower organism to higher organism. And this protein has been shown to be involved in regulating multiple aspects of RNA metabolism. In flies, there is only one muscle blind. In human, there are 3 different isoforms. And this protein has been previously linked with another neurodegenerative disease, which is called myotonic dystrophy. But it has never been linked with ALS or anything related to ALS. So we started looking at the literature, what is known about this protein. And one of the papers that was published from Tom Tussel's lab several years back, and they looked for RNA targets of wild type and mutant fat family protein. And FUS is part of fat family protein. There are 3 different proteins which are part of this family. And muscle blind is one of the targets that he published in his paper several years back. So that further prompted us to look for the mechanism, how muscle blind is involved in regulating toxicity. So we first becomes mislocalized in multiple model organisms. So first thing that we looked for, whether muscle blind knockdown is enough to modulate pathological changes that we see in patients and in different model organisms. So we expressed wild type and mutant FUS in primary cortical neurons. And we looked for as you can see, that wild type FUS is predominantly nuclear, whereas mutant FUS becomes distributed in nucleus and the cytoplasm, and which is more obvious if you see the quantification panel. And we found that if you knock down muscle blind, somehow it reduces cytoplasmic mislocalization of mutant FUS. So that was interesting. We also found that knocking down muscle blind reduces FUS positive stress granules. Mutant FUS goes to the cytoplasm and it localizes into, incorporates into cytoplasmic stress granules. So that was an important finding for us. So then next thing that we started doing was we wanted to know how it is relevant to human patients. So we got iPSC lines that was, sorry, it was, there is some change here. That was expressing a mutation, P525L, in these iPSC neurons, and these lines were generated by a lab in Germany, Jared Stanker's lab, and he differentiated these iPSC lines into motor neurons, and when he knocked down muscle blind in these iPSC neurons, he found that the first positive stress granules were significantly reduced in response to knocking down muscle blind. So further translating our findings from Drosophila to mammalian system to human iPSC neurons. So then we wanted to know whether knocking down muscle blind is enough to suppress toxicity associated with mutant FUS expression. So we have a system where you can express fluorescently tagged version of wild type or mutant FUS, and you can track these individual neurons over time. And if you express mutant FUS, these neurons, they die over time. So knocking down muscle blind was able to suppress toxicity that was associated with expression of mutant FUS. So mutant FUS expressing neurons, they die, whereas if you knock down muscle blind, they survive a little longer. So now we wanted to know the impact of knocking down muscle blind on the morphological features that occurs in when you express these proteins. So R521C expression, it leads to a defect in dendritic branching that is evident from a dendritic elongation. And if you co-express knock down muscle blind in these primary cortical neurons, morphological defect is also rescued. So now we wanted to know that what is the mode of suppression, what is being rescued. So one of the component of cytoplasmic stress granule that was reported previously was a spinal SMN protein, and SMN protein has been linked with SMA. So we looked at the levels of SMN in our cells expressing either mutant FUS alone or mutant FUS with a muscle blind knockdown. So we found that expression of mutant FUS, it reduces the levels of SMN RNA and as well as protein, and when you knock down muscle blind, the levels are restored in response to knocking down muscle blind. So now we ask this question, whether upregulating SMN itself either by genetic approach or by another means, whether it will rescue toxicity. So here, quickly I wanted to show you that when you overexpress SMN protein, you can suppress toxicity. By knocking down SMN, you enhance toxicity. So that further supports our idea that muscle blind rescues the levels of SMN, and if you overexpress SMN or supply exogenously, you can rescue toxicity. So that was a paper which is in press right now, and it will be coming out pretty soon. So we are further trying to identify what are the molecular pathways that are being regulated and being rescued by muscle blind. So that story is ongoing, and we hope to understand the mode of mechanism of muscle blind mediated suppression in near future. So then the second part of my talk, I wanted to talk about extrinsic factors. What are the factors? So first we talked about genetic factors, but one of the components that we are studying is traumatic injury. And traumatic injury has been linked with ALS or ALS-like pathologies in the literature. And when you look at the epidemiological reports, there are reports suggesting that TBI is one of the most common cause of neurodegeneration among sports people. The NFL players, they are 4 times more prone to develop ALS or ALS-like symptoms. where our soccer players, especially Italian soccer players, there are series of reports that suggest they are 6 to 11-fold more prone to develop ALS or ALS-like pathologies. So war veterans, like Pandey suggested, they are 2 times more likely to develop ALS as compared to general population. So we decided to investigate the relationship between traumatic injury and neurodegeneration. So here is a video that I hope it works. And so I downloaded it from YouTube, and I found it very fascinating that these players, when they are on ground, they bump each other. They have a strong helmet. And despite of that, they develop profound neurological symptoms. At least subset of them, they develop dementia, they develop ALS or other types of pathologies. So we tried to recapitulate something similar by taking fruit flies in a tube. Thank you very much. I think you got my message. So this project was started by an undergrad who came to me from Carnegie Mellon with some bioengineering skills. And currently, that researcher is here at Northwestern. He works at, as a graduate student in Dr. Morimoto's lab, Rogan Grant, and he helped me in developing these devices that we are using in our lab. This is a manual device, and this is an automated device. I'm not going to spend too much time on this. I want to jump on the data. So this is the Drosophila Lifecycle, males and females, and they go through different developmental stages that takes about 10 to 14 days to reach here. And we took every developmental stage of Drosophila, adult, first instar, second instar, third instar larva, and then we started giving them mild and repetitive trauma. And we were interested in, and we looked for how many animals they die when we give them a repetitive trauma. So we found that when you give repetitive trauma up to 70-degree angle to the adults, they about 5 to 10% animals die. Rest of them, they survive despite of giving multiple hits at 70-degree hits. So we decided to follow up on these animals. And we found that if you take so we used transgenomic lines where we have endogenous stress granule proteins which are tagged with GFP. So we started giving them traumatic injury at different degree, and then we looked for induction of stress granules. And we found that these animals, if you take out their brains, just giving them traumatic repetitive injury, they show induction of stress granules. And these stress granules, they persist over time. Whereas if you take flies and heat them up, that induces stress granules. Those stress granules, they start disassembling over time. So the granules that you are getting in response to trauma, they appear to be different than heat-induced. So we looked for various markers like ubiquitin as well as endogenous stress granule markers, and we found that these granules, they co-localize with not only ubiquitin and P62, but also it leads to TDP43 mislocalization in response to just trauma. And here you can clearly see that in adult as well as in larval brains, there is mislocalization of TDP43, and it co-localizes with cytoplasmic granules in response to trauma. So now we thought, let's see what are the pathways that regulate these granules. We decided to induce autophagy in these animals, and we found that there is a dose-dependent rescue in the formation of stress granules as well as the mortality that we see in response to these traumatic injury. So now, so far, we talked about control animals. So now what we did was we put ALS-causing genes like FUS and C9, and we started giving these animals traumatic injury. And what we found is we looked for their mortality index as well as their motor dysfunction. And if you give them traumatic injury, you somehow their motor function is further compromised as well as they survive for less time. How much time I have? 5 more minutes? That's plenty. Thank you. So now, what is the mechanism? What happens in response to trauma? So we took these animals, flies, and we gave them trauma, and we decided to take out their brains. And then we did unbiased proteomic analysis. We just looked for the whole brain proteome. And using that approach, we identified about 2,000 proteins that become altered in Drosophila brain. And then we started narrowing it down. So one of the proteins group that came up and they stand out is nucleoporin proteins. And these nucleoporin proteins, they were massively upregulated in response to traumatic injury. So we individually validated a subset of these nucleoporins by qPCR as well as by Western blot whenever the antibodies were available. And indeed, these proteins, they become upregulated in response to traumatic injury. So now we validated these findings in a rat model, and then we found exactly same alteration in these nucleoporins. So now question is, what is the mechanism? How these upregulation of these nucleoporins, they lead to TDP43 mislocalization. So we took individual nucleoporins and we started upregulating them in vivo as well as in larval brain as well as in adult brain. So if you overexpress these NUP62, one of the protein that we identified in our proteome analysis, it leads to TDP43 mislocalization itself. So we looked for TDP43 localization as well as solubility, and we found that upregulating these nucleoporins, it leads to alteration in TDP43 localization and makes it more insoluble. So this story is really long, and I am going to probably stop over here, and with the thought that these nucleoporins have been reported to be involved in C9-mediated ALS, and there are series of reports showing the link between nucleocytoplasmic transport defect as well as in C9 ALS. So we are now doing modifier screen to take these hits, and then we are putting them in flies and giving them trauma to identify if we can identify these some of the genetic modifiers of traumatic injury, as well as in other neurological diseases. So with this, I would like to thank my collaborators, Dr. Chris Donnelly, as well as Dr. Evangelos Kiskinis, who is here, and for offering me support with IPSCs, as well as Rogan, who started this project. I think he's sitting in the back, and Eric Anderson and Suklin Kaur, who did most of the work on muscle blind. Thank you very much for your attention. I'll take any questions.
Hi, that was excellent. Thank you so much. One question about the model you decided to study FUS in. It looked in most of the metrics that you used, like the overexpressing of wild type FUS as a mild phenotype. And I'm wondering if you considered using the Drosophila ortholog of FUS and doing knock-ins, and what the pros and cons are of the approach you chose. Thank you.
That is excellent question. Thank you, Beth. Drosophila version of human FUS is called cabeza. And cabeza means something that is enriched in your head. That's a Spanish word. So we have a model of a cabeza. And if you, and the residues which are mutated in human patients in FUS, they are conserved in cabeza. So we created equivalent mutation in cabeza. And when you overexpress cabeza, we see similar symptoms in flies and similar phenotype. Even probably the phenotype was more profound as compared to human FUS. So we do have that model, although we haven't published it yet, but we focused more on the human version of the protein, but it does lead to similar symptoms.